Hello, President Downey. My name is Kung Lee. I'm Vice Secretary General of APSR 2022. This is my great honor, and thank you very much for this interview. So first question is, please introduce yourself in advance.
My name is Greg Downey. I'm currently the president of the American Thoracic Society, and I am a professor in the Department of Medicine and Pulmonary Critical Care at the University of Colorado, and I'm executive vice president for academic affairs and provost at National Jewish Health in Denver, Colorado. And it is my pleasure to be attending APSR and also to participate in this interview. Thank you.
Thank you. Can you please give a brief explanation of topic and contents that you will share in upcoming APSR 2022?
Yes. So the first talk that I'm going to give is a talk that looks at the role of extracellular matrix and how certain proteinases, and specifically matrix metalloproteinases, alter that matrix. And that leads to inhibition of the ability of the epithelium to repair itself under conditions of acute lung injury. And the practical implication of this is that many people who have the acute respiratory distress syndrome, or ARDS, fail to resolve the injury in a timely manner. And if those patients cannot resolve the epithelial injury, then they tend to have a much more prolonged course of illness, more days on the ventilator, and they have a very much higher mortality rate. And we have been focusing on how the injured epithelium repairs itself under those circumstances. And what our research has shown is that there is a subpopulation of airway epithelial cells that probably reside in the small airways. They express the marker keratin 5, and these cells produce an enzyme called matrix metalloproteinase 3. And that enzyme degrades the extracellular matrix and causes it to become disorganized and also mechanically rigid. And those signals from the matrix are transmitted to the epithelial cells, and we think that those signals inhibit repair. And so what we are working on is ways to antagonize that system, to try and selectively inhibit MMP3, and also to help the matrix return to its baseline state so that the healthy epithelial cells can then proliferate and repair the damaged alveolus. So in a nutshell, that is the first topic that I'm going to talk about. The second topic is one that I think is near and dear to all of our hearts in the last 2.5 years, and that is the topic of severe COVID-19 pneumonia, and acute lung injury associated with that infection. And what I'm going to talk about there is the pathogenesis of the disease, so how the virus accesses the epithelial cells, how it moves from the upper airway to the lower airway, what cells in the lower airway and lung parenchyma does it affect, and then what is the body's immune response to that. And like many other types of acute lung injury, sometimes the body's immune response is counterproductive and actually contributes to the injury. And I'm also going to touch briefly on the role of injury to the endothelium that we know that the SARS-CoV-2 virus can perpetuate. And then I'm going to contrast ARDS associated with COVID-19 to ARDS that's not associated with COVID-19 to see if there are any similarities or differences in those two circumstances. So in a nutshell, that will be the topic of the second talk.
Mm-hmm. Thank you very much. Actually, this is our great honor to have you in our symposium. So your lectures both are very important and very valuable. So we really appreciate it. My third question is, what kind of studying you are currently working on? Can you please briefly introduce your area of expertise and current focus of your studies?
Certainly. So I'll give you two main areas that my lab is interested in. The first one is in the pathobiology of pulmonary fibrosis. And we have been very interested in a pathway that is activated by TGF-beta in fibroblasts and myofibroblasts that then signals those cells to turn into myofibroblasts and lay down excess collagen. And we became interested in this area because we were interested in how the fibroblast interacted with the extracellular matrix, and you can see that there's a common theme there. And in the areas that the fibroblasts contact the extracellular matrix, there are structures called focal adhesions. And in those focal adhesions, there are a variety of proteins, integrins, for example, but also signaling proteins. And many years ago, when I was at the University of Toronto, we discovered that there were several types of tyrosine kinases and several types of tyrosine phosphatases that resided in those focal adhesions. And I won't go through all the details, but there is evidence that excess activity of tyrosine kinases or inhibited activity of tyrosine phosphatases can actually drive the fibrotic process. And we've identified two types of one type of tyrosine kinase and one type of tyrosine phosphatase that seem to be critical in the regulation of this pathway. So the tyrosine kinase is called SRC, SRC, and they're a family of SRC tyrosine kinases that have been implicated in fibrosis. And then on the other side of that equation are the tyrosine phosphatases, and we've been interested in a tyrosine phosphatase called protein tyrosine phosphatase alpha. And we've made genetically modified mice that are deficient in protein tyrosine phosphatase alpha. And also we've made a conditional knockout so that we can delete it in certain cell types. And if you delete PTP alpha from fibroblasts, you actually protect the mice against pulmonary fibrosis in several preclinical models. And I will go into detail in some of the talks, but protein tyrosine phosphatase alpha actually activates SRC. I know it seems a bit counterintuitive, but there's an inhibitory tyrosine residue on the carboxy terminus of SRC. And if that phosphate is removed, SRC becomes active. And so the other part of this pathway that we've been working on is looking at the effect of SRC inhibitors, so small molecule inhibitors. And we've identified one called saracatinib that was originally devised in oncology, but we found in preclinical models that saracatinib is very effective at preventing pulmonary fibrosis in mice. And it is currently in a clinical trial in humans. And so we may be able to present some data from that clinical trial. It's still blinded, but it's a phase 2 clinical trial in the use of saracatinib in the treatment of pulmonary fibrosis. So that's one of our main projects. The other project that I've briefly mentioned before is looking at the role of these keratin 5 positive basal epithelial cells that we think are important in the pathogenesis of acute lung injury. And as I mentioned, those cells we have found to produce MMP3, excess MMP3. And we've seen that both in mouse models of acute lung injuries. So for example, influenza infection in mice causes these cells to proliferate, but also we've seen the same thing in human lungs. And we have the ability to obtain explanted human lungs that are not used for transplant. And many of those patients die from acute respiratory failure and actually meet the clinical criteria of ARDS. And so we've used some advanced proteomic and genomic technologies, single-cell RNA-seq, for example, and we have discovered again that there's this population of keratin 5 expressing cells that produce this enzyme MMP3, and those are vastly expanded in the lungs that are injured as compared to the lungs that are histologically normal. So those are the two main topics of research in my laboratory at present.
Thank you very much. My last question is about your advice for APSR 2022. So can you please give us your request and advice for APSR 2022?
Well, first of all, I am really looking forward to joining you in Seoul at APSR. And I look forward to it in several ways. The first is to meet my colleagues and friends from around the world who I haven't seen for many years. The second is to meet new colleagues so that we can understand what we're doing in research and clinical research so that we can collaborate and share information, but also potentially actually collaborate in research. And thirdly, I really look forward to interacting with trainees. One of the things that I like best in my professional life is learning from young scientists because their minds aren't so fixed. They can look at things broadly and they don't have all these preconceived notions. And I love discussing scientific problems and clinical problems with them. And then lastly, I am looking forward to the culture of Korea. I love Korean food, and I greatly look forward to interacting with my colleagues, both old and new, in Seoul. So that's what I am really looking forward to.
Thank you very much for your valuable comments. Also, this is our great honor to have you in our symposium. So hope you meet in APSR 2022, and thank you very much.
Okay, and I look forward to seeing you in just about 3 months, I guess. So great. All right, thank you very much.
Thank you.